All right, now the big one. Everybody hates this one, including me. Okay, so oxygen hemoglobin dissociation curve. So first an intro, because I think that always helps. All right, so you have this problem. Oxygen isn't particularly soluble. So we can't carry many grams of oxygen in our blood because there isn't enough water, right? We would have to circulate many hundreds of gallons of water to deliver that much oxygen, which doesn't make sense. Okay, so we need to deliver more grams or more volumes, amount of oxygen, right? So remember, amount is not concentration. Concentration is a ratio. Amount is an amount, right? So over on the right side of that graph, volumes is amount of oxygen. It could be grams, it could be volume at a particular temperature and pressure, but in any case, it's amount. So amount of oxygen, right? Now there is still a PO2, right? This is the oxygen dissolved in the blood. Okay, so now we get to talk about hemoglobin, the molecule. Hemoglobin is a protein. It's a 4-subunit protein. Each subunit has what's called a heme molecule, which is this complex ring structure, okay? Hard to break down. In the middle of that heme structure is an iron. That iron can hold one O2, not an O, but an O2. So it means that our hemoglobin molecule, which is like a square with 4 quadrants, our hemoglobin molecule can hold 4 molecules of O2, right? Now, red blood cells are essentially bags of hemoglobin, right? That's all they are. Like they're just chock full of hemoglobin. So each hemoglobin can hold 4 oxygens. You have lots and lots of hemoglobin in each cell, and you have lots and lots of cells. Okay, so you have a lot of places to put oxygen. And when you take oxygen and stick it on the heme, it isn't dissolved anymore. All right, like it's not playing a role in PO2 anymore. Not playing a role in PO2 because it's stuck to hemoglobin. All right, so we have this idea of saturation. If you have a lot of hemoglobin that each has 4 binding sites, what percentage of those binding sites has oxygen attached? That is saturation, right? So when we talk about 100% oxygen saturation, we're not talking about PO2 at all. We're only talking about hemoglobin. We're saying that all of the binding sites on all of the hemoglobin have oxygen attached, okay? So that's one state. At the 0 saturation point, that means all of the hemoglobin is empty, right? And at 50, half of the hemoglobin, half of the binding sites have oxygen attached, okay? So that's this axis, that's this y-axis, is the hemoglobin saturation, how much hemoglobin is carrying oxygen. Now, along the x-axis, we have partial pressure of O2, like we've been talking about, dissolved oxygen. PO2 is going to vary. In the lungs, it's high. In the tissues, it's low, right? So as the blood moves around the body, it's going to vary. Well, empirically, experimentally, if we examine hemoglobin, if we look at how the saturation changes over the partial pressure of oxygen, this is the curve that results. Okay, in other words, if we, so like at a partial pressure of 50, hemoglobin is 80% saturated. At a PO2 of 80, hemoglobin is 90-ish% saturated. Okay, so this is experimentally determined. Like, it is a property of hemoglobin. Okay, now why is it important? The 2 different shapes, we have a steep part and a flat part. Okay, in the steep part, when PO2 is low, it is fairly easy to increase saturation for a small change in PO2, right? So this slope says that a small change in X makes a big change in Y. If we move up here to the top end, at high PO2s, we see that a big change in PO2 makes only a small change in saturation. So we have a molecule that behaves differently in low oxygen areas than it does in high oxygen areas, right? That's essentially what this inflection point is showing you is, okay, when we get to having lots of oxygen, hemoglobin is sort of greedy. When we have very little oxygen, hemoglobin is very charitable. It dumps oxygen very easily, okay? So it says that hemoglobin will bind oxygen where oxygen is high, okay? So like in the lungs where the PO2 is high, the saturation is high. So if hemoglobin comes in and it's the PO2 around it is 40, okay, down here. Well, in the lungs, it is very quickly going, because at the lungs, the PO2 is like 100, right? So we're going to go from 40 to very close to 100 relatively quickly because of the high PO2 in the lungs. So when PO2 is high, hemoglobin grabs on. When PO2 is low, we see that the saturation falls quickly for small decreases in oxygen. Short version is this state, this characteristic of this molecule allows it to gather up oxygen in the lungs and effectively dump it in the tissues. So it makes it a transporter, and one that is somewhat active. The cool thing about hemoglobin is it doesn't require any ATP to release or add oxygen. So it means that the only thing that has to change is the partial pressure. So if we move hemoglobin to an area where there isn't much oxygen, it drops its it drops its oxygen into that area that needs it. So it's a very intelligent molecule. So it also means that that oxygen is only released in areas where there's low PO2. So if we take a molecule of hemoglobin and we move it through a tissue that isn't busy, well, the PO2 in that tissue is going to be relatively high. Hemoglobin's not going to lose much oxygen. That's good, that's efficient. We want it to drop its oxygen in the places where the PO2 is low because that's where it's needed.